And so today we have with us Daniel Manrique Castano. He's the creation officer and at the Digital Research Alliance of Canada. Daniel received his PhD in neuroscience from Ruhr University Bochum in Germany, focusing on rodent models of stroke and glial biology. He's passionate about statistical modeling and research data management and stewardship. After completing his postdoctoral fellowship at Laval University in Quebec, Daniel joined the Digital Research Alliance of Canada as a researcher, data curator for the Federal Research Data Repository, the FRDR. And in this role, he promotes fair data practices, metadata standards, and long-term preservation strategies to researchers across disciplines. I'd like to remind you all that if you have any questions, please type them in the chat, and we'll go over them at the end. And with that, Daniel, you can take it away.
Cool. Thank you very much. I'm pleased to be here sharing this space with you. I will now try to share my screen and verify. So is it working for everyone?
Yep, perfect.
Oh, perfect. So thank you very much for being here to share this space. Today we will be discussing a little bit of how to handle and sharing, how to handle biomedical images and sharing reproducible workflows. And this is a resource I have been developing and encouraging in my current role as a curator officer in the Digital Research Alliance of Canada. So we kind of start by the agenda. We will mainly focus on 5 points: understanding, planning, gathering, organizing, and sharing, which are more or less the roadmap we researchers usually have when doing bioimaging. So from all the methods for collecting bioimages, doesn't matter what's the method we use. The image data analysis and data in particular is the core principle to everything. So we need to handle our images and our material thinking in how are we going to collect, to handle, and finally to analyze the data. And the problem we face today as a researchers is that we collect a huge amount of data. We are talking about terabytes, petabytes of data, which for many of us is difficult to handle, especially when you are PhD and postdoc, for example, and you need to have all the imaging data you produce, for example, in your personal laptop or in some external hard drive that most of the time are not up to this task. So it's important to, from the beginning, to think about proper handling of those images and proper managing as well to build a reproducible workflow. This is a scheme of the bioimage life cycle that we can perhaps start from the phase of planning, how I'm going to acquire my images, ending by the long-term archiving and sharing of those images. I forgot to mention this presentation is completely available online. It's a GitHub repository, so if you scan this QR code, or at the end you also will see some links to access the presentation. You can also check all the resources which are here highlighted with a blue link, so you can check all of it thereafter. If we think about planning and acquiring images, there's something very essential that we face all the time, and it's basically full hard drives which are not capable of managing our images, and most of the time we are extremely happy in front of the confocal microscopy, in the wide field microscopy, acquiring images, but then we don't think too much about how are we going to store, to move, especially, and to document, which is a task that we as a researchers usually do not care about, but it's extremely important for reproducibility. And of course, how to analyze our images. Right now, the research landscape, specifically in Canada, is changing a little bit. And perhaps you are aware that now laboratories are required to submit research data management plans, which basically describe how the information, the images collected during the study will be handled. We think that this will, in the future, in the very near future, evolve towards an scenario where we all researchers need to share our raw data. In that case, we will need to share our images, and we need to start thinking about how to do that. No, so uh, the first aspect we need to consider, of course, is the imaging hardware that we need to answer our research question, but also in terms of the storage of images, what's the necessity we have, thinking about the complexity of the images we take, that can be in the order of gigabytes or terabytes, could be single or multiple focal planes, etc. And when you do data acquisition, we always hope to find an equilibrium between the parameters I use in the microscope uh, and the needs to answer my research question. So of course, as neuroscientists, we will love to take all our images, full brain images, are at 40x with innumerable details and very beautiful stain cells, but sometimes we need to think about how to deal with those images. And I'm sure for you will not be alien when you took images of whole mouse brain slices at 40x, and then you will open them in ImageJ and you will realize that it take 20 minutes to open, or perhaps your laptop cannot handle those images, etc., and will never open. So that's something to think about from the beginning, and we are used to do not think about from the beginning. Also, prioritize open file formats like TIFF over the proprietary ones like CSRI from Zen. And very importantly, keep always record of the metadata. And we are going to talk about metadata a little bit after, but it's a key component of handling images and producing definitely reproducible science. So this is an example of the kind of brain images I made during my research time, and you can see that from the screen it's exactly the same image, but it has, for example, in this case, a different bit depth. One is taken at 8 bit and the other one is taken at 16 bit. And it's exactly the same image. And it's funny because when you work with researchers, and I know many of you are in this situation, when you make an image at 16 bit, the first thing you do when you open ImageJ is to transform it to 8 bit, because ImageJ and the plugins with ImageJ are uh, much compatible with 8-bit. So the question is, why do I take an image of 16-bit, that's the double size, if at the end the first thing I will do is simply to change to 8-bit? This is a very small aspect, but I know that all of you deal with it. So this is the kind of thing that as a parameters we need to start thinking. I can give you another example. From my perspective, in the stroke field, an image at 20x doesn't offer you more information than an image at 10x, for example. If your objective is to count cells, for example, like in this complete brain slide, a 5x is enough rather than 20x, which it's more space, more acquisition time, etc. So of course there's no a definite guide to define parameters for imaging, but I know you are aware what I'm talking about. Simply think about what do I need to answer the research question, and what's the parameters I can choose, I can select to answer them without overwhelming with a space in my laptop and with a huge amount of data that perhaps I do not need to answer my research questions. So basically, I like very much this phrase that a microscope is as good as the person that operates it. And before doing imaging, before handling a microscope, definitely it's necessary that we as a researchers get sufficient training to be able to define good parameters for our imaging. I mentioned at the beginning that it's better to use open formats like TIFF rather than proprietary formats like CSRI or LIF, which is from Leica. And there are open and free tools available to transform those files. Bioformat is perhaps the most common one, and it's compatible with Fiji. Also, it has a Python instance. There's what is called a new generation format, which is the OMT format, which basically keeps all the metadata a CSRI image has. For example, you will perhaps know that when you save your CSRI image as a TIFF, the size of the image is lower, and the reason is because the TIFF doesn't save all the metadata contained in a CSRI image, so the total size of the image is lower. But this OMT file, it keeps the complete metadata, the complete record of how the image, so the parameters used to take the image. It also support multidimensional imaging. You can see beautiful examples in these links. And actually, TIFF formats they have a limit because of the way the format itself is built, and the OMT format basically breaks this limit and can work with much bigger images than the TIFF itself. There's a new format coming, which is called the OMSAR, which has the properties of an open file like the TIFF one, but also the properties of a proprietary file to handle big images. You will notice, for example, that if you do a microscope image and you will open it in Zen, for example, if you took the image using um, a Zen microscope, then you open the Zen Blue software, the image opens faster than if you use, for example, ImageJ, because size in the software has what we call image pyramids, which basically doesn't load the whole image directly, but small sets of that image. And when you zoom in that image, it basically renders data that was not rendered before in the whole view of the image. So that's called a pyramid. ImageJ bioformats from ImageJ usually doesn't work with pyramids, but proprietary formats like CSRI and the Zen software does. So what OMSR is now bringing is the same amazing, faster renderization of images, but to an open format similar to TIFF. So here you can explore these links to see many examples of the OMSR format and how it works. I think that in the very next few years, this will be the gold standard to format of image to work with and also to share and you can handle it from graphical unit interfaces like Fiji, or programmatically, not going to Napari or to Python, etc. So it's amazing indeed. Most of the images rendered by the Allen Brain Institute have already this format. So till now, the take home message I want to transmit is that the planning you do from the beginning and the data management is crucial, and thinking about platforms to store and handle your images and formats, it's a must from the beginning of the project. And for example, the alliance, the organization I work for, it's one of these platforms that can help you to really handle and finally host your images. At the end, from my perspective, this is not the alliance speaking, this is Daniel speaking. I think data is more important than articles. Basically, articles are just interpretations of data, but the most crucial, the most essential artifact we can offer to the research community as a researchers is data, and this data should be ideally reusable by anyone, because we are using public funds, no? Gathering image metadata. So most of the images we produce are images that can be understood by us, but very rarely they could be understood for by other scientists or by person outside your research project, no? So we have challenges with image metadata, which is basically metadata is information about the data, is data about the data. And in the field of bioimaging, metadata basically refers to all the parameters that we as scientists employ to acquire the image. And we have challenges, which is basically that we have a fragmented ecosystem that lacks clear standards that guide us as a researchers on how to collect what metadata is important and how to share it, and especially journals and institutions lacking uniform guidelines. And I would like to make a strong call here, and is that in terms of scientific quality, in terms of the thing we are talking today about, which is handling bioimages and sharing reproducible workflows, we cannot wait, or we cannot expect uh, institutions and specifically journals do something for us. So we cannot wait until journals ask for raw data, until journals ask for specific metadata standards, because they will never do. The journal has a business, and the business is to make money. Sharing data, sharing metadata, reusing data is still not a business for them. So that's a thing we as a scientist need to be aware about and really encourage from our position as a researcher. So we have the image that journals are the quality gatekeepers of science, but they are not. They make money with public funds. That's it. So the quality of science needs to be directly from us. And I think I will make a very quick exercise here. If you go to Nature Communications, for example, which is one of the journals that ask for data, if you submit a publication there, we can open, for example, whatever, it doesn't matter, this about macrophages. So if you click there and then you go here to source data, then here you see the source data. You can download this stuff to your computer, and then you open this. You will see that all the data which is shared here, it's completely unusable. It's simply void data that scientists share to check a box into the submission. But if you are more or less familiar with data, you will know that these tables are not usable because they are not in tidy format. They are not documented. So this is tables that you can see, yeah, here very nicely, but these tables information nobody else can use because it's not tidy, it's not um, documented, so it's useless information. And then let's see what we have here. So here are images. And if you know a little bit about imaging standards, you know that PDF is not a format to share images. And for Western blot, we would like to have the original, perhaps stiff PNG image that the device, the Western blot device, showed to us, not a crop made by a researcher. So this is really information which is unusable, is not useful for anybody. So we need to take care about that because journals will not. And you can do the exercise you want with Nature Communications because I made this exercise always and never found or get surprised that there's data well documented or data I can use as a researcher. It's always the same thing. So it's basically useless data. So specifically for the biomedical field, we have a group of recommended metadata that basically says a group of parameters that are the minimal aspects we need to report, the parameters we need to report on how we acquired our images, and those parameters are of use for a broad spectrum of researchers and are minimal to understand and to reproduce perhaps the experiment. So here you find links to inform yourself about these parameters, and this is a template that is offered also. You can download here in this link, and basically it talks about the type of study you performed, the kind of biomedical sample you handled. We are talking about the organism, the variable, the genetic background, etc., the experimental conditions, the compression, the dimension, the size, the type, the voxel size, the pixel size, etc., the spatial temporal alignment, all that stuff. And that's something you don't need to write yourself in the keyboard or know by hand. That's data which is embedded in the metadata of your image. If you took an image using a size microscope, a Leica microscope, 95% of this information is embedded within the image. You just need to be aware that the information is there and to extract that information from the image. We will see in a minute a really nice tool, for example, on how to do that. For the metadata, it's important to use controlled vocabulary, and this is example of ontologies. This also are links that you can explore ontologies to name in a standardized way from organism to experimental factors or imaging methods. For example, wide field imaging. I don't know if it's called, like, if that's the standard name of wide field imaging or confocal imaging, or it's called laser confocal imaging. All those terms standardized are available in these ontology websites, and I recommend you to explore to know how to name the variables and things in your uh, from your bioimaging experiments. These are a couple of examples of web-based ontology services. This is from Europe. I think this is American, the two of them. So if you want to know how what's the standard name of a microglia, of an astrocyte, of XY microscope, you can go here and you will have standardized naming conventions, which are still extremely useful when you want to share data and actually when you want to find data from other researchers. So this is an extremely useful tool to record the metadata specifically from your imaging device. It's completely open and free. So it's called the MicroMeta app. So you can Google it, and you can go to the presentation, and here is the link, and there's an associated research article explaining how MicroMeta app works. Actually, I think there's also some YouTube tutorials by the person who developed the software to explaining how to operate it. And what this does basically is allows you to record the parameters of your imaging device. In this case, a microscope. You need to fill in at least one per experiment. For subsequent experiments, you perhaps needs to change the objective, the exposure time, etc. But I found it quite useful because if you track all the metadata from your imaging device, and then you use this other plugin called Methods G2. So the MicroMeta app and the Methods G2 plugin are are a single unit, are assembled, the two of them. So when you record the metadata, Methods G2 plugin will generate for you a text explaining what are the parameters you used to acquire your images. So that's something in that case you do not need to do manually as we often do for research articles, but the methods G2 taking the file, the configuration file you have generated for your experiment using MicroMeta app will generate the text for you, and that helps you to save time in one hand, but more importantly, to be precise and sufficiently enough informative of what were the parameters you used to generate your images. So there's a GitHub repository, and as I said, a research article that can help you to understand a bit better how this work, and I highly recommend this approach to gather metadata from your images and device. If at your institution in McGill or from your lab or the Alliance, we also have, I know that the Alliance hosts several OMERO instances for researchers in Canada. OMERO is one of the best options to handle and to recover metadata from images. It's basically a web service client that allows you, let's call it like a kind of Google Drive or Dropbox, but for images online, which does a really good job extracting metadata in different formats, and it's an amazing interface to do handling of your research images. So basically, you will not be transporting images across your lab, your research institute in hard drives that can easily break down, get lost, etc. But all your images can be simply hosted in OMERO, allowing access to all the people that needs to have access to the images in a very simple way. So I highly recommend to check OMERO if it's available in your lab, institution, department, etc. Or if not, I know that the Alliance can host several OMERO instances for researchers. And this is a list of metadata you can consider at the moment of handling your images, at the moment of documenting your images, the complete list, and insightful explanation. It's available in this link in this research article. But some examples are, for example, the channels, the annotation, um, the position, the zoom, the range, the dynamic range of the image, etc. And as I said, you don't need to know this by heart. All this information it's embedded in your images, and you can extract it in many ways. So I invite you to check this list in your in the article and gather as more information about your image as you can to document it. There's no hard guidelines for the moment, but be, better documented. It's is the best documented possible is the better we can do. So in terms of metadata, we need to move to better consolidation of standards, like to help researchers to define or to provide them with better guidelines on what information do we need to make my images understandable and reusable by others. We need to develop tools like MicroMeta app or the Method G2 plugin to take metadata from images and make it available for the public. The companies like Leica or Zeiss, they do record this metadata, but most of the cases, this metadata is not human readable. If I open the metadata from a CSRI image, I will have literally tens of thousands of parameters that as a researcher I can hardly understand. So the idea is to work together, hopefully, with these companies to take a minimal, highly desirable metadata package that whatever researcher or potential reuser can understand. And of course, if we think at the institutional level, we need to think about infrastructures to support the proper handling collection of metadata records. So if we go about organizing and sharing our biological data, we have talked about the extremely huge size a data set, especially in the neuroscience field, can take. And most of the time, they are in the laptop of students and postdoc. I will bet an 85% of the time they are here in the laptop you are using to attend this conference right now. There's some other spaces like the Institute network, perhaps the cloud like Dropbox, Google Drive, which of course are not ideal places to store scientific, highly relevant data. And when we think about storage solutions, we need to think about making the data as open as possible. So basically, we want the data to be in a safe place, and that the more the a good amount of potential reusers can access this data. There's many options we have available as researchers. These are just some of the available repositories. Perhaps Zenodo is one of the widely used by the community, but also in McGill you have an instance of Borealis, and especially I will highlight further because in further we are kind of specialized in handling big data sets. Perhaps in OSF, in Zenodo, in Borealis cannot storage for you 10 terabytes of data, 20 terabytes of data, but we at Further, we can do that. And it's a service which is free for researchers. I mean, it's not free, it's pay with public taxes, but it's available without cost at research labs. So if you go to this table, you can see some of the characteristics of many of these repositories. Some of them are paid depending of the storage space. Some of them are free all the time. But for Canada, we have Borealis and we have Further, which are a couple of great repositories to work with. If you want to, both Borealis and Further are Gerenalis repositories. So that means that we receive all kind of data, but if we think about repositories dedicated specifically to imaging data, I invite you to think about these two, which is the IDR and the Bioimage Archive, uh, which are IDR, I think it's American, Bioimage is European, which are repositories which handle specifically bioimage data and also have very nice storage capabilities because being repositories dedicated to bioimaging, they know how big bioimaging data sets can be. So I invite you to explore also this couple of resources. There are other solutions, as I mentioned, OMERO. It's a really nice web interface that different to the other services I share. OMERO needs its own instance. So basically, that means you need to have your own space somewhere in the cloud to host an OMERO instance. And as I also said before, I think the Alliance can offer you support in that regard to host an OMERO instance. And there's some other open science related, let's say data management related platforms like IROTS, which also can be an interesting resource to host images on the web. So what are we expecting? What will be like the minimal desirable requirements of when we share images as a researchers? We would like to see the raw data and also the processed images. We would like to see images which are not compressed or in open file formats. We would like to see the metadata, for example, using the OMTIF format, which is a great example of open format together with metadata explaining how the image was acquired. PNG images are extremely useful if we share, for example, the images we use to illustrate a research article. PNG is preferable. Importantly, images should have accurate and descriptive naming conventions. And that's, for example, something I work with researchers when they submit data to Further. I help the researchers make each of the image has a unique identifier that as an external person, as a potential reuser of the dataset, I can tell the difference between one image and other image just based on the image name. And naming conventions could sound like a trivial thing to do in research, like how to name your files, but it's not trivial at all. And most of the time, we scientists, we have very extremely really bad naming conventions for our files, being images or not. And I do that because I work with researchers all the time. README files are essential, and README files perhaps are familiar to you when you install a software. The software at the end of installation always asks you, do you want to check the README file? Do you want to see the README file? And the README file for an image, it's basically a plain text file that explains the nature, context, and content of the images, and it's crucial to understand what the data set it's about. And also I encourage you to use CC BY or CC0 licenses, which basically allows the broad public to have access and to reuse your images. I'm sure you have seen in research articles, when you go to the data availability statement, perhaps 90% of research articles, they said data will be shared upon request. Guess what? They are colleagues, they are scientists. 90% of the time, the data never appear. I have been reviewer of research articles, even being a reviewer, where I ask for the research data, the raw data, and the data never appears. When you see this statement are available upon request, most of the time, 90% of the time, the data will not appear. And you can do the exercise yourself. You can email 20 scientists, 20 research papers that says that the data will be available upon request. And if you are lucky, you will get one or two links to very bad formatted, not understandable data sets. And that's not the idea. The idea is to make our research transparent and available to anybody. Sharing reproducible workflows. With reproducible workflows, we think about mainly three aspects: the data being objective, reliable, and reproducible. And from a research data management perspective, when we analyze biomedical image, we have access to a big asset of data sets. We need to have records of how the images were processed, and we need to share our results. It's extremely typical that when you address research article, it's extremely hard, or even you cannot understand how the data was extracted or how an image was processed. I recommend not to explain in your method section how the image was processed. Simply share your code, your annotated code, and that will be much descriptive than an explanation in a research article. So when we think about image analysis, I invite you to consider a modular approach, which is basically handle the images as independent subtask that you can use each module as its own completely independent component. So we can think of two different levels in the image analysis workflow. Low level, which is not in terms of if it's important or not, simply a naming convention. When we talk about the low level, that means technical knowledge, basically the technical instances we employ in our images, which is the processing and the analysis of images, object detection, segmentation, particle tracking, etc. That's called low-level technical knowledge. And we also have the high level or the disciplinary knowledge, which allow us to interpret, to understand, and to interpret the data we extract from images. So always think about these two instances. A complete scientist perhaps has the two. If you have a extremely deep disciplinary knowledge but not technical knowledge of how to handle images, perhaps you need to pair with a colleague which has the technical knowledge, etc. But always thinking in the two components. This is an schematic of a typical image analysis workflow. We have the images, and we can go throughout an established workflow. We can create a new workflow, or we can implement a machine learning workflow, and basically to get any input we want. And to do all the processing of those images, there's a very interesting list of software that you can use. from Fiji to Galaxy. Here you can access the links in the presentation to all these instances um, to process your images. And the core characteristic um, of these applications listed here is that all of them allow you to provide a reproducible pipeline of the steps you have taken to process your images, and providing the raw Fiji script, the raw cellpose file, or the elastic file is much more informative than describing in the research article what was the procedure. This is an amazing resource. I invite you to explore it, especially because it has many models for different software to perform different tasks. If you took a microscope image and you are wondering, how do I count the cells in this microscope image, in this page called bioimaging.io, you will have different pipelines for different software developed by different people of how to do a specific task. So I highly recommend you this database of analysis to explore depending on your research question. As we have standards with metadata, we also have standards with image analysis workflows, which basically are listed here, and that includes checklists for publications ready workflows, for newly created workflows, or for machine learning workflows, and basically entails many of the elements we as a researcher need to share with the public and for potential reusers. And that includes a definition of the regions of interest. We have used the settings, we have used the code, the sequence of a step, the limitations of our procedure. It also could entail indeed a tutorial of how to implement the procedure, etc. So it's a very nice element to take into account. So here also, this is so my first highlight was the think about the journals. Do not wait for journals to ask this, because they will never do until they find a revenue from it. And the other big highlight I would like to share with you is we as neuroscientists need to be capable and competent in using code. And I found this quote, I think it was on LinkedIn, perhaps in Twitter, I don't remember exactly, but I will allow myself to read it. The mouse is antisocial. The GUI is antisocial. So what's that mean? You have a problem to solve and you solve it with a GUI. What do you have? A problem solved. And when you solve it with a command line interface in a scripted environment, you have an artifact. And all of the sudden, that artifact can be shared with someone. So here the message is use code, not the mouse. And I'm not a coder myself, or I was not training code in any instance of my academic career. I did it by myself. And for me, it's we as neuroscientists, we spend a lot of time learning complicated research techniques. I had to learn how to do middle cerebral artery occlusion in mice, or how to open mice, insert a filament, etc. If you people you do patch clamp, you know how tricky, how many weeks, months takes to learn patch clamp. But even if we spend weeks, months learning a neuroscience technique, it's extremely a problem to learn some basic coding skills to analyze data. And I think that coding, I think there's no a strong reason for that. I think that coding need to be um, a core skill into the research, into the professional career of researchers. If we want to do science, if we want to produce data, we need to learn how to handle, how to process, and how to analyze the data. And that's done using code in the current scientific environment. So I invite you all to start, if you haven't, to start a little bit of steps using code. Overall, so in for the image processing pipeline, keep track of all the procedures and changes you have performed. Organize and link your files using naming conventions. So I talk about that naming conventions is not a trivial aspect. And perhaps you can, I also make a strong call to share the final or intermediate results of the processing pipelines you implement during your research. Uh, as I said at the beginning, no, as I said at some point of the talk, sharing data it's extremely useful for the purposes of reuse. And when we submit research articles to these publishers, most of the time these publishers copyright all this information we have produced using public funds. So I will give you an example. I'm writing right now a book about astrocytes, and while looking for material on internet, there's a bunch of extremely nice images in the scientific literature I cannot use because it's copyrighted by a publisher. So I encourage you. We know that all the material we use, if we think about biomedical images, we select a small sample of those images to illustrate our research article. Why not to publish all these illustration images in a repository so anybody can use them? Imagine, you can perfectly imagine a teacher in a high school in whatever part of the world, North America, Africa, Thailand, using your repository of beautiful image neurons to teach students how the brain looks like. And that's something that we scientists are in a very particular position to offer if we can share those images with the public. This is just an example, but this is to encourage you to do not think about just the publication and the citations that publication will have. Think about also in sharing all the material you as a scientist generated in different ways. And I think that the images we do in microscopes are amazing and can help to not just to educate, but for many other purposes in the world, and it is a no cost for us to share it with the community. Uh, Here in this link, you can see some other examples of how to publish images and figures used in a publication. This is from my personal endeavors as a researcher. So basically, from my research or from my latest research, all the images I have generated for illustration or for analysis are public and can be used for anybody in the world. I know that a person I know from the United States in Colorado, a teacher in Colorado, is using them for the small module of neuroscience that he uses with his students. So it's a nice thing to do. I talk about this. So share your data and do not trust this data is available on request. Share your data as openly as you can and keep it as closely as necessary. So sharing data is not only an exercise to meet the journal requirements or the founder's requirements. I believe it's truly an ethical and professional responsibility, and this is to ensure reproducible science and facilitate access and reuse of research data. So I think that research need to move towards competent researchers in research data management and data analysis. Data analysis, it's another complete different story. And if you invite me again to Open Science Hour to talk about data analysis, we can talk about data analysis. Also use standardized approaches to sharing raw data and analysis code to support our findings. And this will make researchers committed to transparency and best practices. These are the two most important Canadian repositories. Further, the repository I work for, but you also at McGill have Borealis, which is also an excellent instance to share your data. So this presentation is available on the web. This is a resource I invite you to consult specifically to be aware of all the recommended metadata standards of many of the tools to produce reproducible workflows. It's available in English or French, this presentation. Here you also have some supporting material, some training resources from the Alliance. And I'm happy to answer any questions at this point.